Okay, you guys are doing good. Okay. So what we're doing?
So we started out talking about the pathway selection of cholesterol neurons, and kind of drew a little diagram where they were located around the spinal cord, including the midline, and then
Not now, right? That's fine.
you're right, in the spinal cord, and when there's a midline and how foreplate is there, and then in the midline there's a netrin and slit, netrin attractant, chemoattractant and slit, the chemorepellent, and the foreplate has sonic hedgehog and it acts as the attractive signal. Sonic hedgehog and receptor is called patched, and there's patched 1 and 2, and then there's desert hedgehog, you kind of just mentioned that, and then Indian hedgehog. But then these commissural neurons will move down and they'll eventually cross the midline, which is the axon of the neuron will cross. But normally, as it moves down, there's on the neuron it has has patch at the head, and it has the DCC receptor, and then a ROBO1 receptor, and then slit will bind to ROBO1 and activate it to cause blocking of DCC so netrin can't attach to it. And then the sonic hedgehog will also bind to patch on the head of the neuron and activate SMO, which promotes DCC, and these two things working together keep it moving straight down.
Okay. Then what happened?
Then the axon will start expressing ROBO3, which blocks ROBO1, and then it'll also send a signal to the nucleus to make 14-3, which is a protein that blocks SMO, and that opens up DCC for netrin to bind to it, which causes the axon to move across the middle. And then as it gets there, ROBO3 downregulates and 433 goes down. ROBO1 is then unblocked and blocks DCC again, and SMO is activated to move away from the center. A slight problem in this process can result in a large coordination issue with the eyes.
Okay. Then what happens?
Then we talked about the cell adhesion as a mechanism to travel, and so the axons travel on laminid, and they have integrins and incadherin to stick to the laminid. So they travel on neurolaminid, and they travel in bundles called nerves, and the bundles are formed by fasciculation, and then defasciculation is caused by polycylic acid and is regulated by BMP. When we talk about synapse formation, so synapse forms when an axon contacts a target, and then for the example we talked about the cholinergic neurons, the muscle neurons, so that release acetylcholine as the neurotransmitter vesicles. So at the growth cone, it secretes agrin, which aggregates the acetylcholine receptors on the myotube. It then spreads out and makes, and then is surrounded by Schwann cells that secrete the myelin sheath, and then multiple neurons making connections on muscle fibers in development. But this is refined after birth, and one axon connects to one muscle fiber. And we talked about the Schwann cells, the stem cell divides, some express notch and receptors for notch, and then the notch is cleaved and moved into the nucleus, or moved in, to the nucleus where the neuron, to form the neuron, and then the others form the glial cells.
Okay, good. So now we are moving to the next topic, and we are going to talk about various things of the which form, and those are forming from the mesoderm. Okay, so we are going to talk initially about paraxial and intermediate mesoderm. what is that I'm going to explain it. So this is essentially we are going to talk about praxis and intermediate mesoderm. And so remember what happens, like we have, so this was your embryo, and right here, this was your epidermis layer. So this is your epidermis layer. And right here, you have your neural tube formation took place. We will talk about that. This is your neural tube. And beneath this, right here is your notochord. And then surrounding this whole thing, all this is your mesoderm. And then all this is your endoderm. And this is your archenodome. Okay. Now we know about this thing. And this archenodome is essentially your captive gut. So, if we look here and we draw in a different way, like want to draw from the top, what we see there is, we will see right here, will be a layer of epidermis. So try to draw it a little bit big so we can find out what is happening. And right beneath the epidermis, right here, this is your neural tube. Now this mesoderm, depending upon its location, can be named differently, right? And what happened when we are talking about this embryo like this, so this is the embryo, so if this is your neural tube right here, and beneath your neural tube, this is your mesoderm, right? Now mesoderm is surrounding it, mesoderm is not on the top, mesoderm is throughout like this, right? Now, the mesoderm is there, so mesoderm, depending upon the location where it is, it can be named differently, right? Now, what happens that the mesoderm that is next to neural tube on both the sides, this will be called paraxial mesoderm. Paraxial mesoderm. And then mesoderm next to this. So if you think this is your spinal cord, this is your spinal cord, if you're looking the body from the top, right? If this is the spinal cord, then this is on the two sides of this. Making sense? So this is the spinal cord, so the mesoderm is here, mesoderm is, I'm looking from the top. Okay? Now, this is paraxial mesoderm, then you're moving further, right? Then you're moving further. So then the next mesoderm, which is there, this will be called, so interesting thing you have to think about when you're thinking about, before I name it, you're thinking about the continuation, and then you're thinking about the separation. You have the ribs, these muscles are there, then further you don't have any bone, right? Then you have your pelvic girdle, then you have the bone. You have to think like that. So that's how I'm drawing those, and I will tell you what part of the body they are going to give. Okay? So this is called intermediate mesoderm. And after that, we have another part of the mesoderm. And that mesoderm is kind of like drawn like this. This mesoderm is called lateral plate mesoderm. Okay. Now lateral plate mesoderm is going to give you splanchnic. Splanchnic means the circulatory system. So the circulatory system is coming from there. Then it's going to give you body cavity, pelvis, and limbs. So body cavity, pelvis, and limbs. Now, other than that, so this part is called splanchnic, and this body cavity, pelvis, and limbs, these are called actually somatic. And other thing it is going to give you is what you call extra embryonic mesoderm. Remember when we were talking about the placenta, we said extra embryonic mesoderm come, right? Which will be outside that, and that's going to make the blood vessels of the trophon. That is coming from here. So blood vessel, all the circulatory system, and the circulatory system of the trochoepiderm is also coming from this lateral plate mesoderm. Now the intermediate mesoderm, this mesoderm is going to give you gonads and kidney. They are coming from intermediate mesoderm. Okay, now, so think about what is this thing? So think about this is head and this is rest of the body. Okay? And this is your spinal cord here, and this is your brain. Spinal cord, right? And paraxial mesoderm is surrounding on both the sides like this. So this is on like this, right? Now, this paraxial mesoderm, what is going to give you? Is going to give you the whole, because this is surrounding like this, so this head is coming from the paraxial mesoderm. So paraxial mesoderm is going to give you head. That's the first thing. And then, remember we said here are vertebrae, right? Those vertebrae are bone, those will come from the mesoderm, of course, right? So vertebrae are not a straight bone, they are segmented, right? So this mesoderm which is here, that is actually segmented, that segment. And this segmentation of mesoderm is by what kind of genes we did talk?
Fox.
Hox genes. So this segmentation is caused by Hox gene, and each of this segment, so this segment, this will be called somite. So each of the segment of the mesoderm will be called somite. So then it is going to give you head, and it is going to give you somite. Okay. Now each of the somite is going to give you multiple things depending upon the location in this body axis varies. Okay. Now these somites are going to give you sclerotome, sclerotome is cartilage. It is going to give you synditome, synditome is tendon. Then it is going to give you myotome, which are muscle. Then it is going to give you endothelial cell. Now endothelial cells are lining the blood vessels. So blood vessels then are combination of these two things. So you have this lateral plate mesoderm that give you circulatory system, right? But then you have those endothelial cells which are lining the blood vessels. These are coming from these somites. And then it gave you dermatome, and dermatome is going to give you actually dermis. So dermis is the connective tissue of the body beneath the skin. So all those muscles beneath the skin are coming from these somites. Making sense? So what we are saying, like the mesoderm can be divided into multiple parts depending on the location where they are. And we have basically three different types of mesoderm there. We have right here paraxial mesoderm, intermediate mesoderm, and the lateral plate mesoderm. Lateral plate mesoderm gives you splanchnic, what you call circulatory system, somatic, which is body cavity, pelvis, and limb, and also the extra embryonic mesoderm. Then we are saying we have this lateral plate mesoderm, this lateral plate mesoderm we talk about. Then we have the intermediate mesoderm. Intermediate mesoderm gives us gonads and kidneys. Then paraxial mesoderm, it gives us head and multiple somites, and these somites are created under the direction of the Hox genes. And each of the somite can give you sclerotome, which is cartilage, synditome, tendon, and myotome, muscle. Other than that, they also give you endothelial cells, and they also provide you dermatome, that is dermis, the musculature of the body wall. Sorry. Okay. Okay, so after about a minute, then we'll go further. Any doubt? You're okay? Okay, now let's talk about all these different type of mesoderm, and we are going to start with our discussions with the paraxial mesoderm. Paraxial mesoderm. Now, paraxial mesoderm, if you, I'll draw it here again. epidermis. This is the neural tube. And on this side is your correction. And also the trigger. And right here is your so this is your lateral plate wisdom. This is your paraxial wisdom. This is your neural tube. So this is your paraxial wisdom. Okay, now. So what this paraxial wisdom is going to become, it depends upon the signaling center, what kind of signal it is getting. There are 4 signaling centers here which are controlling it. So we have 4 signaling centers. What are those 4 signaling centers? We have epidermis, epidermis. What epidermis is doing? Epidermis is secreting BMP4, and epidermis is also secreting from here Wnt6. Okay. Then this lateral plate wisdom which is here, it actually also secretes BMP. So 4 signaling centers. Number 1 is epidermis. Number 2 is lateral plate wisdom. And then the neural tube, it also serves as a signaling center. What does it secrete? It secretes 2 things. Number 1, neurotrophin 3. Neurotrophin 3, it's called NP3. And Wnt1. Neurotrophin 3 and Wnt1. It is signaling there. And then the another signaling center you have here is your notochord. So notochord, what happens, I didn't talk about the point of notochord, what happens to notochord. Remember that what is happening here? This is your paraxial mesoderm, right? So notochord is right here under the neural tube. And neural tube is your, this is your neural tube, which is your spinal cord, right? And we said this paraxial mesoderm is going to give you the vertebrae, right here. And notochord is also just beneath the spinal cord, right? So what happens? Notochord disintegrate. Notochord disintegrate and say this is a vertebrae. This is a vertebrae form. Right? This is another vertebrae form. Another vertebrae form. In these vertebrae, you have this spinal cord going on, right? Notochord disintegrate, and notochord disintegrate, and some of the cells of notochord come like this, come like this, come like this, and notochord then form is totally disintegrate, but it forms vertebral, intervertebral, I write here, but intervertebral, write it properly. It forms intervertebral disc. So those cells are there, just form intervertebral disc. So it's totally disintegrate. Whatever is left, that just form the intervertebral disc. Okay. Now, so what is happening there? So notochord is still there. Notochord is also a signaling center. So that's the fourth signaling center. Number 4 is notochord. And notochord is secreting sonic hedgehog and noggin. Making sense so far? Now, so right here is your paraxial medulla. So this is a somoid, right? So you have different area. Somoid. So we're talking about one somoid. Okay. So right here from here, what is happening? This is sending BMP. BMP is there, right? But you have a notochord here. So this is sending BMP. And when it is sending BMP, you have a notochord here. Notochord is sending noggin. And what noggin does, noggin block BMP. So BMP cannot do anything. So noggin has completely blocked the BMP. Okay. Then what happened? Sonic notochord is sending sonic hedgehog. Right? So sonic hedgehog is there, very high sonic hedgehog is coming here. Very high amount of sonic hedgehog. Very high amount of sonic hedgehog and very high amount of noggin. That means there is no BMP, only sonic hedgehog signaling is there. Right? What that does, that causes this portion of the somoid to start expressing PAX1. Okay, so it starts expressing PAX1. Now what PAX1 does, PAX1 activate the genes for cartilage. So, because of that, therefore, this area, it becomes sclerotome. Sclerotome. So it becomes sclerotome. Okay? Now, what sclerotome is doing, expressing PAX1, activating the gene for cartilage. But then it also has to, does not want it to allow to become muscle, right? So what it should do, it inhibits myogenic regulators. Myogenic regulators means the transcription factors responsible for formation of muscle. Okay? And then what happened? The area which is next to it, this area is going to become what we call syndicome. syndrome, and that is going to give you tendon. And we'll talk in detail what happens here in this area later. Okay? Now, area above here, right here, it gives us, now here, if you look at very carefully, look here. This side of this somoid, similarly, this side of this somoid is close to neural tube, right? Whereas this side is away from neural tube. Making sense? So it means we are saying if a neural tube is a signaling center, so the signal which is coming on this side, let me draw this. So the signaling which is here will be different than the signaling on this side. Okay? So, if this is like this, let me draw it like this. Draw the same color. Okay. So the signaling which is happening in this area is different than signaling is happening in this area. Making sense? Now, if you look here, this area is directly above the epidermis. This is away from epidermis. That's why I have drawn the line like that. Making sense? So this area will have more exposure to what is coming from here, and more exposure to what is coming from here versus what is coming from here. Making sense? So, what that does, that actually create a center even like this. Making sense so far? Now, so this area, which is, since they are going to give muscle, so area of the somoid, all of these are going to give muscle. Area of the somoid giving muscle. So this whole thing will be a myotome. Myotome. M-Y-O-T-O-M, myotome. So a somoid is divided in three parts, right? You have this area, which is going to give you the cartilage, right? And then we call it sclerotome. Then we have syndrome, and then we have myotome. Making sense so far? But then in myotome also we are saying areas are different. Now this area which is close to neural tube, this is called primaxial myotome. And this area which is away from the neural tube, this is called abaxial myotome. Okay? Making sense so far? Okay, now talking about this area. The area under this, triangle like upside down triangle, this area is called this color. Okay. So this is called derma myotome. Derma myotome. Or dermatome also. You might read dermatome or dermamyotome. Now, in this derma myotome, the area which is kind of in the proximity of this primaxial or abaxial myotome. Now this area this red area is going to give you dermis. Okay. And the area which is right here in the center, this will always keep on giving you proliferating myoblasts throughout the lifetime. That means these will be the stem cells. And this area, the proliferating myoblasts, they will be always expressing PAX3 and PAX7. Okay, and the area, the abaxial and primaxial, they are giving you different, all the body muscles are coming from there. Every body muscle is coming from these areas. So these area which are abaxial and primaxial area, they are going to give PAX3 expression. So PAX3, PAX3. This is PAX3 and PAX7. So this is PAX1 and this is PAX3, PAX7. So let me go back. So before making it more complicated. Okay. So what we are saying, we are talking about the paraxial mesoderm, and we are saying that paraxial mesoderm is divided into certain pieces, and each of that piece is called somoid. And we are saying that this somoid is experiencing, each of this somoid is experiencing 4 signaling centers. And if we look at the location of these, we are saying that this epidermis is there, neural tube is there, and neural tube on both the sides you have the somoids there, right? So what these somoids are getting? These somoids are getting signal from epidermis, which is BMP and Wnt6. And then these somoids are also getting signal from neural tube, and neural tube is going to give you neurotrophin 3 and Wnt1. And then we are saying that the next signaling center is this lateral plate mesoderm that secretes BMP. And the 4th signaling center is notochord, and notochord secretes sonic hedgehog and node. And we are saying, we did talk about the fate of notochord also. We said notochord, kind of these disintegrate later. It's here now, but later it will disintegrate and it will form the cells of intervertebral discs. Okay. Now, so this notochord is sending the sonic hedgehog, and lateral plate mesoderm is sending the BMP. But notochord is sending the Nogin, so Nogin block BMP, and sonic hedgehog, very high concentration of sonic hedgehog coming here in this lower part of the somoid, and this somoid then start expressing PAX1. PAX1 start expressing the genes which are required for the activation of the cartilage, and this portion becomes sclerotome. And PAX1 is going to inhibit the transcription regulator for the muscle formation. Okay? And then we are saying the area above the sclerotome becomes syntotome, and I didn't explain how that happened. So first we understand this signaling, then we'll go there and talk about that. But we are saying that syntotome is formed there, and then syntotome is going to give you the tendon. Then we are saying the area above the syntotome is called myotome, because that's going to give you different part of the muscles. And now myotome is, if you look here, myotome is having different signaling center, but we are saying a part of the myotome which is closer to neural tube is called primaxial myotome, and part that is away is called abaxial But both primaxial and abaxial, they are away from the epidermis. So area that is near the epidermis, that triangle area, is called dermamyotome. And we are saying in that dermamyotome itself, the area which is closer to abaxial or primaxial, that is going to give you the dermis, the muscles of the body wall or under the skin. And then in the central area is going to give you the myoblasts, those will be the stem cells for the muscle throughout the life, right? And we are saying these are expressing PAX3 and PAX7 transcription factor. And the cells which are present in abaxial and primaxial and abaxial, they are expressing PAX3. So we are saying PAX1 going to give you cartilage, PAX3 going to give you muscle, PAX3 and 7 together, you are getting stem cell-like characteristics. Making sense? And there's more to it, and we'll talk about that stem cell-like characteristics later. So far, any doubt here? Okay, then talk to each other, then we'll go further.
Does the notochord disintegrate as the vertebrae are formed?
Say it again?
Does the notochord disintegrate as the vertebrae are formed or after?
After.
Okay.
After. Because when the bone is going to form, it's going to actually crush up.
Gotcha.
So then they will move, the cells are going to move under those. Any doubt?
Would you say that proliferating myoblasts
Yeah.
are the same thing
The-
as stem cells?
Yeah, for example, when you have when we say myoblast, that means they are committed to become muscle,
Okay.
but they are still the stem cell. For neuron, we call them neuroblast. Okay, so these are proliferating, it means they have not come up the cells like the rest of the body. Remember, muscles on the body.
Okay.
That's why I use the term proliferating myoblast. Any other doubt here?
Okay.
Okay. Any doubt here? Okay. Now let's talk about how the myotome is determined, right? Determination of myotome. So what happens in myotome? So remember, we have this here epidermis, neurothelium, and this is here. Abaxium, and this is your primaxium. Okay. So, if they need to become muscle, right? If they need to become muscle, they need to express to become muscle, you need to express myogenic regulator. Okay. And myogenic regulators are essentially the transcription factor required for muscle feed. Okay, so those are MIF5, MyoD, MRF5, 4, myogenin. And these transcription factors are so strong in their activity. They are so strong, each one of this can activate muscle specific gene in any cell. So you take a cell, you basically put any of these there, that particular cell is going to start expressing the genes which are going to make those cells into muscle. So essentially, by taking those, you can convert any type of cell into the muscle cell. So they are very strong regulators. So what is happening there? So right here, from here, you are getting neurotrophin 3 here. Okay. From here, you are getting Wnt6 here. And also, from here, you are getting Wnt1 here. When you get both, all the three things, Wnt6, neurotrophin 3, Wnt1, you start expressing TAX3 and TAX7. Okay. Now, what is happening here? So right here is your lateral plate mesoderm. So what is happening here? This is your notochord. Notochord, very high sonic hedgehog was there, right? And this is little far away, the primaxial. And what is going to happen? The primaxial is far away, the sonic hedgehog is going to go there, but a low or the weaker, lower sonic hedgehog is coming here. Okay. Now, when the lower sonic hedgehog is coming there, then what is going to happen is, from here, the Wnt1 is going to go there. So what happened? So you have this cell. What this cell receive? The cells here, they receive Wnt1 and they receive very small amount of sonic hedgehog. These two things are coming. Okay. So Neural tube is giving Wnt1. Neural tube is giving Wnt1. And then sonic hedgehog is coming from motor cord. Okay. When that thing happens, what it does, these cells start PAX3 expression. Okay. When this PAX3 expression occurs, PAX3 start expressing myogenic regulator. And in this case, PAX3 induces MIF5. PAX6 induces MIF5. And then MIF5 induces MRF4 and myogen. Okay. And when these genes are activated, so they can, as we said, they are very strong regulator. What they're going to do, they're going to start expression of muscle specific genes. Okay. So what happened here, all the back muscles, they are actually made from primaxial area. Okay. So all the back muscles are going to be made. So what is happening? We are saying that in this, we did talk about dermamyotome. We said in the dermamyotome, so this was the dermamyotome, right? Dermamyotome. In the dermamyotome, now this is interesting. This is dermis, right? So what is happening? In the dermamyotome, you are getting Wnt6 from epidermis, neurotrophin 3 from the neural tube, and Wnt1 from the neural tube. Now, although all these cells, they should start expressing the PAX3 and PAX7. Keep them proliferating. But what we are saying, that the cell here, which are on the periphery, they become dermis, right? The muscle of the skin under the epidermis. Why that happens? That happens because right here, under the expression of PAX3, these cells here, we'll talk about this also in a minute, but in the paraxial area, these cells are starting making MIF5. So MIF5 is here, then other muscle specific genes are activated. So those genes influence these peripheral cells which are here. Remember? So that's why these cells become dermis. Making sense? Now, coming to here, to the primaxial area. Now the primaxial area is away from notochord, not that close as this area was there. Okay? Now, you get the sonic hedgehog signaling, but sonic hedgehog signaling is weak. Very less sonic hedgehog come here. But you have lot of Wnt1. Now, remember neurotrophin 3 is secreted from this area, that goes top. But here, there is no neurotrophin 3. Okay? So, here what is happening? You are having Wnt1 coming here. So, Wnt1 directly going there, sonic hedgehog coming there. They together in these cells start expressing PAX3. When the PAX3 is expressing, PAX3 induces MIF5, and MIF5 induces MRF4 and myogenin, and they further start expressing muscle cell gene, muscle cell specific gene, and these are going to become the back muscles. Now, if this is the area, for example, so it means this cell, this cell, this cell, this cell, this cell, this cell, these cells, right? So these cells are still not called muscle. Muscle is a long fiber, right, with many cells coming together. So these are still called myotome. That means they are still the stem cells. They have not changed into muscle yet. They will change muscle yet later. But here, they are determined to become the muscle. That's what we do in our lab experiment when we throw retinoic acid. We do a determination experiment, right? We say, okay, they have become neurosphere, now they have become determined. So this is the determination of primaxial endorme myotome into the muscle lineage. That's what we talked so far. Making sense so far? So talk to each other for a minute, then we'll go further. Yeah.
Can you just explain real quick again, you were saying that doing myotome has 0.61 and 53.
Yeah.
And that causes them to impacts 3 and 7 expression
Yes.
to stay in
Proliferate.
proliferating myoblasts,
Yeah.
right? And then, but you said the dermis area.
Is the
here,
surrounding area, right.
Yeah, has MIP5.
It does not have MIP5. MIP5 is coming from, MIP5 is made here, right?
Yeah.
So MIP5 activates an MRF4 and other muscle specific. So those proteins are influencing the cells surrounding.
Okay. So influencing the
The
surrounding
edge.
area to
The
become
edge, yeah.
dermis. Okay.
Yeah. Any other question?
No, that was it.
Question? You're okay? So we are still on the determination, but we did talk about primaxial, we did talk about the abaxial, right? So we are going to talk about what is happening in the abaxial area. So again, the same diagram. Neural tube, notochord, lateral plate, mesoderm, and so we have talked about these two, what is happening there, right? So now we are going to talk about the abaxial. So abaxial is going to give you 4 limbs and ventral body wall muscle. So 4 limbs are coming from there, and all our front muscles are coming from there. Okay. So what is happening there? So right from the epidermis, you have Wind 6 coming here. Okay. And when the Wind 6 is coming there, this lateral plate mesoderm is also there. And what that is doing, that is secreting a lot of BMP. Right? Now BMP which was coming here was inhibited by Nobin. Right? So that we said we start making PAX1 because all the sonic hedgehog was coming here. So we had a lot of sonic hedgehog here. But here, Nobin, which is here, cannot reach to this abaxial myotome area. Okay. Now, if it cannot reach there, then BMP is not going to change it into the muscle. Okay? So you need to inhibit, what time it is?
10. 10.
10? We have 5 minutes.
Yeah.
Yeah, so BMP need to be inhibited. But Nogin cannot go there. Actually, in this abaxial myotome area, there are cells present in it, and that which are Nogin secreting cells. And this is interesting. These Nogin secreting cells are made in epiblast. Made in epiblast. They express Nogin in epiblast cells. So when I'm saying expression, I'm saying gene expression. But no Nogin protein. Okay. During development, these cells migrate to abaxial myotome. So this is interesting. So think about these cells are made right there in epiblast. They show the expression of Nogin, but they don't express the protein. So all those talks about gene expression actually have no meaning, because gene expression doesn't make any sense unless until the proteins are not there. So all those talks of having same gene expression doesn't go much forward in terms of function of those genes. Function of those genes is determined by once you have the protein. But that protein here is made when they are coming here in the Nogin secreting cell. And that's what we have been talking, like all the mRNA is stored in the egg, right? But that mRNA never expressed till the fertilization takes place. So now, BMP is blocked, Wnt6 is coming there. Okay, so what happened? That these cells, they are getting Wnt6, BMP blocked, and these cells start making PAX3. And when these cells make PAX3, what they do, they activate MyoD. They activate MyoD. And MyoD activate MRF 4, 5, and myogenin, and they activate muscle specific genes. So what we are saying? We are saying that abaxial myotome, it receives Wnt6 from the epidermis, and it also receives BMP from the lateral plate mesoderm. but that BMP needs to be blocked. And that BMP is blocked by the cells which are already present in this abaxial myotome, and those cells can inhibit, they secrete Nogin to inhibit the BMP. We are saying these cells are actually made in the epiblast, they express the gene for Nogin, but they never make the protein Nogin. And these cells migrate to the abaxial myotome during the development, so they stay there. So now we have BID6 coming there, BMP blocked, so these cells start making PAX3. In this cell, PAX3 induces the expression of MyoD. In the primaxial, it was MEF5, right? It's a different. Here it is MyoD. And MyoD activate MRF4, 5, and myogenin, and they further change these cells into the muscle-specific gene. So you have now abaxial myotome, abaxial myoblast cells there. So these are becoming abaxial myoblast cells. Now these cells are going to give you muscles of the forelimb and the ventral body cavity. Now, if these cells in experiments actually they have done, so they have done the experiment where they have removed these Nogin cells, not completely, but a lot. So those cells have very weak ventral body muscle. You can actually see their heart and everything herniated out. So all these things are kind of herniated out. So they don't have very well muscle quality. So talk to each other for a minute, then we are, I think we're then done with this particular. So we'll continue on the
When we talk about the epiblast cells and how they show the Nogin expression, there's no protein.
There's no protein.
Is there no protein until they get to that bacteria?
Yes.
So
They
there's
make that
still.
only when they are in the abaxial.
Okay.
It's the same thing, like when we said when the egg is warm, it has all the mRNA. So you know, breast seed has it, the vector is already there,
Yeah.
right? But it doesn't make that protein until
So,
the process of it, right? The same thing.
okay. Thank you.
Any question here? You have any question, Abir?
No.
Okay, then we are good to go for the day and we'll meet on Monday.
Dr. Moore, I won't be here Monday.
You'll not be here on Monday?
No, I'll be in Texas.
Okay, no worries. So tell someone to record it and email you the recording. And when you come back and we can talk about it, okay?